Why is data visualization complex when interpreting brain maps? Imagine trying to read a map of the entire brain, but instead of roads and cities, you see millions of tiny points, lines, and colors all representing different neural details. That's what interpreting brain maps often feels like. Brain imaging data are enormous, often reaching terabytes or even petabytes in size. This means visualizing such vast amounts of information requires powerful computers and clever techniques to display it clearly. The brain is made up of many different types of data. You have the shape of the brain, the connections between neurons, activity patterns over time, and even molecular details. Combining all these into one picture is tricky because each type of data needs a different way of showing itself. If you try to display everything at once, it can become confusing or overwhelming. Then there's the brain's complex structure. Its folds and grooves, called gyri and sulci, make it hard to tell where one part ends and another begins. Sometimes, tissue samples can have tears or folds that distort the view, adding more confusion. Plus, every person's brain is slightly different, so scientists have to use standard maps to compare data across different individuals. Brain activity isn't static either. It changes from moment to moment depending on what a person is doing or thinking. Visualizing these changes over time adds another layer of difficulty. Researchers need tools that can show how connections form and dissolve, or how activity moves across regions in real time. Another challenge is making sense of the data's uncertainty. Measurements can vary, and scientists need to see how confident they are in what they observe. Using colors or scales to show this variability without confusing viewers is a real balancing act. Finally, as artificial intelligence and machine learning are used more in brain research, visualizing their results becomes even more complicated. These models produce high-dimensional, abstract data that don't fit neatly into traditional brain maps. Creating visual representations that make sense of these outputs takes new and creative approaches. All these factors mean that visualizing brain maps isn't just about making pretty pictures. It's about handling huge, diverse, and dynamic data in ways that help scientists and doctors understand what's happening inside the brain. This ongoing challenge drives the development of smarter, more flexible visualization tools tailored to the unique demands of brain diagnostics and research.